Get started, I wanted to bring up an idea and get your thoughts on this. One of the big things with this class, and something that I've been trying to harp on, is the ability to evaluate, synthesize the data. So, here's what I'd like to do, actually. We've got an exam coming up in a week and a half, 2 weeks. I don't know exactly the date off the top of my head. But, here's what I'd like to do. I want to break the exam into two parts. I want to do a 25-point, 1 point each multiple choice. That's in class.
I like that.
Okay? And it's going to be, do you know, do you understand, maybe a little bit of analysis, but it's more just knowing and understanding what we've talked about. 75 points, take home. And you can do it in one of two ways. 3 ways, actually. You can handwrite, you can type, or if you want to, you can talk the whole thing out. You can voice thread the whole thing. You're not taking it, so I don't really care about it. Damn it. I guess I don't know.
I'm not going to take it.
So, I just let that sit for a day or so. Let me know what you think about it. But one of the fears that I have is that a lot of people are like, I don't know what you want us to do. I want you to do a problem like that critical thinking that we did for Wednesday. But I don't think it's reasonable to ask you to do that just on point in a 2-hour block period of time. I think it would be better to say, you've got your resources, you've got your notes, because that's how the real world works with this. Right? So that's what I'd like to do. What's that, Jonah?
That took me a lot longer than 2 hours, and I had like the full power of Google.
So that's why I'd like to do a take-home for it. That's why I'd like to do a take-home for it. Instead of having you guys have to sit here and stress out over trying to figure out what the f does he want us to do.
Yeah.
Okay? So unless there are any big objectives to it, and please let me know if there are, but that's what I'd like to do, because I think that's kind of probably the most realistic to the real world than anything else. Okay? I see a relief on a couple of faces, but I can't tell if that's just it's Friday or what. So
It wouldn't be like
That's right.
reading the paper like we did today, right?
I don't know. I thought that might be an option, but we'll see. What I will probably do, and I will actually put up in the next week or so, I will put up some old exams so that you can have an idea of what have I done in the past, so that you can start to get a feel for what these would look like. All right. No, I don't think I would force you guys to do a paper, at least on the first one. It's still too early. We haven't had enough reps with it. All right. Bring samples Monday. Bring samples Monday. And
Everyone to bring a sample?
pair. And actually, just so you know, I isolated like 50 phage the other day. So we got lots of extra. So I'm not telling you. I'm not telling you where I got it from, because that's for you to figure out.
What if I bribe you with like a bag of coffee?
No. You got to do a little bit of thinking. Remember, this is like a 1,000 point assignment. Either you succeed or you fail in finding phage. So.
No, that was only if that's the only thing.
All right. Good point, Morgan. All right. So, just on Monday, drop off your stuff in here, and then just head over into the lab, and we'll kind of get started once everybody gets there. I'll make sure to have as much as I can set up. Take a look at the protocol. I've put it up on Blackboard. It's under the phage project tab, and just so that you have kind of a rough idea. I understand that not a lot of it's going to make sense, but there are at least enough people in this classroom that have done this once to be able to kind of guide others. So, like, I think, Bill, you're going to help me out with this, right? Okay. So we'll be able to get everybody covered and help out with it. Before we get into the paper, I wanted to bring up one last technique which they do use in this paper. That's real-time PCR. And we talked about how we'll talk about reverse transcription PCR on Monday, I'm hoping. But the one thing with this that I wanted to point out is we said there's limitations to PCR. What are those limitations? Not quantifiable. Why? What typically happens near the end of a PCR run? Grace?
It's out or like it flattens out.
Yeah, it flattens out. It looks something like this, right? Where early on we don't have much to detect, and then we've got this really nice amplification phase, and then we have a phase where it kind of flattens out. And we said that this is probably due to a number of different reasons. Polymerase becomes less active, or DNTPs are becoming limited, or we also said the byproduct of the building DNA, those pyrophosphates, can build up as well and inhibit the reaction. So real-time PCR, what this ends up doing is taking a snapshot after each round of replication to tell you how much DNA is there. The technique that we use here, there's two major techniques that are used. One's called TaqMan. Personally, I prefer that method because it has some additional power. And there's less optimization that needs to be done. But the technique that we have here set up at Edgewood is CyberGreen. And CyberGreen works a lot like ethidium bromide. So if you've talked about ethidium bromide in the past, it's non-fluorescent normally, but when it intercalates into DNA, it changes its structure enough so that it will fluoresce under UV light. That's how we can, when we're looking at DNA gels, see those DNA bands. We're typically visualizing them with ethidium bromide. CyberGreen works in a similar fashion. Instead of hitting it with UV light, though, we use a laser light to produce the color. So in this process, we perform a traditional PCR. We use primers, we use Taq polymerase. Typically, the products for real-time aren't going to be larger than a KB. Maybe for CyberGreen, maybe 600 to 800 base pairs in total length. The CyberGreen reagent itself is not fluorescent when it's not bound to DNA, but once we start to get amplification, it intercalates into the DNA and now fluoresces. And what happens is we have a camera basically looking at our individual PCR reactions, and it will take a picture after each cycle. And it will tell us what's the fluorescence in the well. So then, obviously, at the end of the run, we have much more fluorescence than we do at the beginning of the run. And what you end up getting then is a curve like this. Oftentimes, we're not looking at the absolute amount of fluorescence in each cycle. We're actually looking at what's the change in fluorescence. How much has it increased from the previous cycle? So from cycle 8 to cycle 9, for example, it's gone up from 110 to about 175 or so. Okay, so the change from 8 to 9 is 175, and from 9 to 10 is 250, and so on and so forth. Okay, so that represents the amount of change in fluorescence from one cycle to the next. We end up getting a curve like this. How do we analyze this? Becomes the next question. I don't remember if I have this in the slide or not. Nope, I don't, so I'm going to have to draw. Oftentimes, we can use computer-based programs to help us analyze these results, but there's a pretty simple way. What you're looking for is the following. Let's say that you have let's say we have this one right here. What a computer program would do is it would draw a line through the most linear portion of the curve. It's identifying this point at being where amplification is its most robust, or being at its greatest efficiency. Where that line intersects, or where the y value is for that, or sorry, the x value for that is what we call a CT, or a cycle of threshold. This is a number a number of cycles that it takes to reach this threshold or this amplification point. Right. Yeah. So the computer first draws this threshold line, indicating this represents maximal amplification. And then if you just draw a line straight down from that, that X point, or straight down to the cycle that represents your CT value, or cycle of threshold. Okay? So in other words, in this example here, it takes 8.5 cycles to pass that threshold. Okay? Now let me draw another part to this, and you can start to tell me some things. Here's another curve. Here's another one. The CT is being drawn at the same point through each of them. Or, sorry, the threshold is being drawn at the same point through each of them. Okay? Still right here. But what differs between the three of them? Which cycle, or that CT value. So if we were to, again, kind of draw a line straight down, this one, its CT value would be about 6. We said this one was about 8.5. And this last one here is, oh, I don't know, 11.5 roughly. Or H.5 apparently. Okay. Go ahead, Jonah.
I'm sorry, I think my brain
No, that's okay.
for a second. What's the threshold value again?
So the threshold value again is it's usually computer generated. It's a point where it's identifying that, there's a couple ways to describe it. One, usually at that point is where you have maximal amplification of your DNA, or most linear. The other way to describe it is it's looking for a linear portion or a linear line that can pass through the linear portions of each of your curves. Oftentimes we're doing these with like 96 individual curves, and so it's going to draw a threshold line based on those 96 curves.
I don't remember anything from genetics, possibly. I believe, for instance, there's something about background fluorescent noise or something. Like, is that?
You will see that, but honestly, this is kind of a small example. Usually the background noise occurs at like cycle 35 to 40. So it's much, much later in the process. And that's a good thing, because that says that the background signal is representing a minor percentage of the overall signal. Yeah. Angela?
So then what do you do with the CT?
Yeah, that's the next part, is what the heck do you actually do with this? There's a couple of different ways to analyze this. Ultimately, what we want to know is, what does this represent? How much difference is there between the amount of DNA in this middle sample, the blue line here, versus the one that is this purple line, okay? And oftentimes what we will use is just kind of there's different ways to do this. You can make it more specific, but the simplest way is to calculate what we call a delta CT measurement. What this means. We said that the cycles it took for this blue curve, 8.5. We said for this one it was 6. Which one has more DNA to begin with? This one. We're seeing the curve show up sooner, okay? So what we would end up doing, and one more thing, for every one copy of DNA in PCR, how many do we theoretically get? Theoretically get 2. Conservatively, if you're asking me, I'd say 1.8. That's just kind of a conservative measurement. So this delta CT method, what we end up doing is saying the difference in the amount of DNA is 2 to the 8.5 minus 6. So it's 2 to the power of the difference of these CT values. How long it takes for them to pass the threshold.
Can you say that again?
Yep. So in measuring this, we say we know that DNA amplifies 1 to 2 every single cycle. So the difference in the starting amount of DNA between the sample in purple here and this one in blue would be 2 to the difference between their CT values. So 2 to the 8.5 minus 6. Jonah?
So this is comparing so if you're quantify a sample, would you need a control to like compare it to?
You need a standard curve.
Ladder kind of
Yes. No, not a DNA ladder, but a known concentration of sample that you could do tenfold dilutions. And actually, that's exactly what is done. So if you wanted to know, this delta-delta CT method is really good to compare between two samples and say, hey, there's fourfold more in this sample than this sample. That's great. It's semi-quantitative. But if you want to make this a true quantitation, there are X number of copies in this sample, you need to include a standard curve. And so what you would have is a standard curve where you have multiple of these curves, one for each dilution of your sample. You would plot it on a graph of CT value versus known copy numbers, come up with an equation and use that equation to solve for each of your unknowns. Okay? And that's what they actually do in this paper. They say that they know a certain number of copies of norovirus genome per microliter or something like that. That's what they're doing, is creating a standard curve to quantify it. Grace?
So, just to clarify, I think you said earlier, the lower the CT, the more DNA.
Yes. Yeah, and that maybe should make some intuitive sense, right? So, like, if I have, I don't know, 5 apples, and I double the number of apples every hour, and you have 1 apple, and you double the number of apples every hour, how many hours will it take for each of us to accumulate 100 apples? Like, that's kind of what it is, if we're talking about apples. But this is oranges, so, Sabrina.
So this curve has the curves that each dilution, and then you compare your curve to the standard curves?
Yeah. Yeah, exactly. Exactly. Angela, and then Jonah. It does, only in the sense of, usually what you want to do is, if you're trying to look at, how do I want to say this? Yes, it does, and it matters in terms of the comparison. So if we did it this way, right? This is 2 to the 2.5, which is somewhere around, I don't know, 6-ish, maybe a little bit less than that. But that says there's 6 times more DNA in one sample over the other. And this is where it becomes important. You can't say that there's 6 times more in this blue sample. That's incorrect. You could say there's 6 times less. That's more correct. But in putting it in this order, where you put the one with the larger CT value first and then your other one second, you can say there's 6 times more DNA in this purple sample than there is in this blue sample. That's why it's important. So usually what I tell people is, you know what, just calculate it either way that's best for you, but make sure that your answer fits with what you observe in the curves as well, or within the CT values. And honestly, if we wanted to know, you can always just very easily do the inverse here. So 6-fold more, the inverse of that is, well, there's 1/6 the amount of DNA in this sample than there is in this sample. Does this make a little sense? Okay. All right. So that's a really long way of, a 20-minute way of saying, hey, they're going to measure DNA in this experiment. This is basically how they're doing it. Okay? Let's see what we can get through. If we don't get through all the paper today, so be it. But let's see what we can get through over the next 30 minutes or so. All right. So introductions. Norovirus. Naked, icosahedral, plus single-stranded RNA, causes gastroenteritis. Basically, this is stomach flu. Okay? pass by the oral fecal route, highly contagious, can survive weeks outside of the host. Fun fact, and this is about a week old now, 158 cases reported by last week at the Olympics. The whole, I think it's either the Swedish or Norwegian team, has come down with norovirus as of last night. Go USA.
Go USA.
So, those are the background facts on this virus. Let's start out here. This is actually from the supplemental material to explain this first figure. Now, this first figure is a doozy, right? You don't need to actually understand very much about it. It's more about the design of the experiment. So, I think the first question to start with here is they're using a technique called CRISPR-Cas. This is kind of like if you were to think of a pinup model for science right now, this is the pinup model for science. Okay, this is the one that everybody's like, oh my God, CRISPR-Cas, right? What?
Is it Cas9?
Yeah, it's the CRISPR-Cas9 system. So this system does what? What is the goal of using this system? Go ahead, Annie. Knockout genes. That's all they're doing. They're creating complete knockouts of genes. The cells and they've got to create, since this is a diploid cell line, they've got to create 2 knockouts in each of them. So they take these BV2 Cas9 cells, and in each of them, they put in a different, what we call guide RNA, but basically that guide RNA is targeting one of the 30,000 or so genes that are within the mouse genome, generating this multicolored population, and what those colors are to represent that each one is distinct from each other. Each one has a different deletion in it. They expose all of these to norovirus.
I think it was around 80,000 guide RNAs.
Okay, 80,000 in total? Thank you.
Different ones, yeah.
So they take all of those 80,000 clones, they expose them all at once to norovirus. If norovirus is still able to infect, the knockout of that gene has no effect on viral replication. In other words, that gene is not essential to support viral replication. What cells are going to survive? Go ahead, Annie.
The ones without the CD300.
So in this, they'll talk about the ones without the CD300. Let's maybe just start out real quick. Say, what is CD301F involved in? What is its purpose in respect to norovirus? Attachment entry. So if you knock out a gene for entry, or if you knock out a gene for attachment, can norovirus still infect? No. In other words, in the presence of norovirus, that cell line, the one that lacks that particular gene, they're going to survive. Okay? So they're using it's almost like a form of negative selection here. They're trying to find the organisms that are most fit to survive in the presence of norovirus. To be fit, you have to lack the genes that support viral replication or viral life cycle. Jonah.
Does this Cas9 knock out genes in the chromosome, or does it just knock out mRNAs?
Oh, crap. Chromosome. These are chromosomal deletions.
Okay.
And that's how they're being used in this paper. Yeah. Sabrina.
What do you mean by like knockout?
Complete deletion of the gene. So meaning like you have the genomic sequence and now the Cas9 system basically cuts that piece of DNA, throws it away, and splices the remaining DNA back together. So that's what we mean by knockout. As opposed to a knock-in, where we'd be putting in an insertion. So those are kind of typical terms used for these types of experiments. So only those cells that survive norovirus infection are those that have, presumably, a gene that's important for the infection. So there's an enrichment of the cells that can survive the infection. That's what this heat map here, or this STARS score, represents. Basic way to read this, the higher the score, the more frequently they found that gene knockout cell line surviving norovirus infection. Put otherwise, when infecting these BV2 Cas9 cells with CW3 mouse norovirus, they more frequently found that cells lacking CD3001F, they've more frequently found these cells than they did find the cells expressing TMEM30A. Okay. Go ahead, Sabrina.
Did you say with the higher score? Can you repeat that?
Yeah, so the higher score just means that they are finding these, after exposing to norovirus, these cells are more abundant. Cells lacking CD3001F, can we just call it the receptor? Cells lacking the receptor are more often found than those with a gene like this TMEM30A. Okay.
I thought you had to have that receptor for it to infect.
You do,
Oh.
but without it, those cells survive the infection. Right? So it basically, by knocking out this gene, these cells here, the ones lacking this gene, have become, in some ways, naturally resistant to infection. And so when exposing them to norovirus, they're able to still grow. So it's backwards to how you would think about it.
I did that.
I butchered this.
That's okay. That's why we're going through it. All right. So here's where they look at infection. So let's go over some of the terminology on this graph real quick. Obviously, hours post-infection means how many hours after exposure to norovirus. What's LOD? They define it in one of the figures, if I'm not mistaken. Yes, it's a lot. Yeah. Limit of detection. What does that mean?
Like
Break it down, guys.
first be detected above background.
Exactly. In other words, the techniques that they're using, they can't detect any more than 100 plaque-forming units of norovirus per milliliter. Exactly. The smallest concentration they can actually physically calculate, given the techniques that they're using. PFU per mil, so they're looking at plaque-forming units on the y-axis here. This is a technique that we talked about. Note the scale, though. Log base 10. So, I always feel like I've got to cover this real quick. But this basically means, so if you have a log base 10 value of 3, simple way to read these, just put a 10, oh crap. Put a 10 in front of each of these. 10 to the 3rd, 10 to the 4th, 10 to the 5th, 10 to the 6th, 10 to the 7th, and 10 to the 8th. In other words, they're able to calculate 1,000, or 10,000, or 100,000, 1,000,000, 10 million, 100 million plaques per milliliter. This is not atypical for microbiology and virology, because oftentimes we're concerned not with 50% changes, we're concerned with tenfold or more changes. Okay? So this is not an atypical way of looking at this. Oh. All right. So I'm going to step aside for a second. What I want you to do is tell me what is the ability of mouse norovirus CW3. What's its ability to infect the wild type cells, those that lack lack the receptor, or those that lack the receptor but now they've put in the receptor back into the cells. So they've done a knockout and then a knock in the gene back into the cells, restoring hopefully normal phenotype. So take a minute, talk it over with somebody else. Try to describe what's happening in this figure.
Without the 42 hours.
doesn't.
So,
hasn't
are they planning out like a,
Like this.
I guess is it like a lawn of the cells and then like very few platforming units?
It's kind of, yeah, it's similar to that. So what they would do is they take their,
I did that with the CW.
in this experiment, what they would do is they take a plate of the cells. They would add a certain amount of norovirus, so
Without the
10 to the 4th per mil roughly.
delta.
They would then at 12 hours and 24 hours harvest the media from those cells and then do a plaque assay, basically like on a lawn of human cells to then detect how much virus there is.
Is the antibody. At least that's what I got.
That's why I initially saw PFU.
And then the S is
Where are they measuring PFU?
So you can do plaques with human viruses. It doesn't work for all viruses. Sometimes you have to do different measurements, like what's known as CCID50, which is a cell culture infectious dose 50. What's the concentration that causes infection of 50% of the cells? And there's different ways to measure that.
even like simple.
All right. So somebody take a stab at this one. What are the observations? Now let's just start with the wild type cells, or the BV2 cells. What happens over 24 hours? They get infected. How much does the concentration of virus increase over that period of time, Jonah?
1, 2, 3 fold. 4 fold.
3 log fold,
Three.
or 1,000-fold, 1,000-fold, or you could say 3 logs would be fine too. 3 logs. All right. How about when CD 3001, 301? No, 3001. F is knocked out.
Nothing. Or it dies.
Decreases. In other words, what would our interpretation of that be?
That CD300 is necessary for the spiral life cycle.
That's probably the most broad, and that's probably the best one, given just the context of this problem. If we also knew, which we do, going into this paper, that this is a cell surface protein, you could make a more detailed argument saying it's not only needed for the life cycle, it's actually needed for attachment, entry, maybe uncoding, one of those steps where you might see interaction. What about when they knock it out, but then they knock this gene back in, so these blue representatives?
The infectivity is restored.
In other words, what they're showing is that the knockout and knock-in here, what they're showing is that the genetic manipulations that they've made, the effects they're observing, the inability of this virus to infect these cells is not caused by additional genetic mutations. It's caused simply by the knockout of this CD3001F gene. This is not an uncommon technique, especially these days where we're doing experiments where we are trying to manipulate the genome. Doing a knockback, trying to knock out a gene and then knocking back in is a nice confirmation that the genetic observations or the phenotypic observations you make are caused just by that single gene and not other mutations that you're creating. If it had been caused by a different mutation, we'd still see something like this in the knock-ins. I don't know if they're expressing it from a plasmid or if they're doing a knock-in like a Cas9 knock-in. I can't remember off the top of my head. Either way would be fine. The putting it in on a plasmid and having it just express from that plasmid would be the much easier method to do.
Wouldn't you say that's what they did for 2E? Because they're talking about vector or whatever?
Yes, vector is another word for plasmid, absolutely. So that is how they've done it. All right. Sabrina.
So the point of the knockback in is to determine if it was like exactly the knockout that caused the.
And not some other genetic manipulation that they've made on accident, some artifact of the system. Exactly. All right. This is the only reason why Xander's here today, is for this slide. So they ended up using a technique known as transfection. in human cells, what transfection basically means is we're taking DNA or nucleic acid, in this experiment RNA, and we're trying to deliver it to a cell. When I was working with EBV, we would often take 3 plasmids, ones that encoded the GAG, the PAL, and the ONV gene from modifications of human retrovirus. We'd stick them into cells, and then we'd get out retroviral-like particles. Right? So this is a way that you can deliver DNA or genomic material into a cell. This actually bypasses, in this experiment, it bypasses the need for a host receptor. So here's how the technique is done. DNA or RNA, backbone is negatively charged from the phosphates. And we add a reagent known as lipofectamine is the most commonly used one. Basically, it's got a hydrophobic tail, hydrophilic head groups. So it forms a mycel. And this mycel-like structure will form an envelope around the genetic material, enter by endocytosis, and eventually we get fusion between the endosome and the lipoplex that they list here. And then that DNA hopefully doesn't get degraded, but rather gets transported to the nucleus, for example. Okay? So this is the technique that they're using to bypass the need for a receptor altogether. Go ahead, Jonah.
Would like a gene gun, electroporation, or heat shock be considered?
Electroporation is actually an alternative that would commonly be used. However, electroporation, it's really touchy. So some cells, lipofectamine works better. For some cells, electroporation works better. It's incredibly tricky to figure out just by saying, yeah, I'm going to choose this technique. Oftentimes that's what we do. I'm going to try this technique. If it doesn't work, I'll try the other one. Yeah. Annie and then Amelia.
Does it also have to do with the size of the genome?
It can, yes. So typically, if I'm not mistaken, electroporation I think works better for larger, yeah. Whereas this lipofectamine or heat shock in prokaryotic cells usually works for smaller plasmids. Amelia?
You said that this avoids what? Like
So it
the-
avoids the need for a host receptor,
Oh, okay.
right? Because in this case, in this experiment, all they're doing is delivering the genetic material of the virus into those cells. And then they're going to test, well, can the virus actually perform its life cycle? Okay. So really what they're trying to do here is they're trying to tease out what step of the viral life cycle is CD3001F required for. Results are on the right. So, again, looking at plaque forming units, I'm guessing this is after 24 hours or so after the transfection. Wild type cells, those that have the gene completely deleted or those in which they've knocked it back in. Observations. No significant difference. They've even run the statistical tests to support that. In other words, by bypassing attachment, entry, and uncoding, by going straight to biosynthesis, they have allowed for this virus to still infect these cells. Supporting what? What does that mean about this particular protein or gene? Jonah?
That the CD300 protein is like responsible for attachment?
Absolutely. That's a great argument given what they've observed. Now, why does this work? Why does this work for this virus? It's a single-stranded RNA virus, and single-stranded RNA viruses need what? Ribosomes, its own polymerase. They're not delivering the polymerase here. They're just delivering that genomic material.
Because it's plus.
What does that mean?
It means it's directly translated and that it produces it.
Exactly. This is a unique feature to plus single-stranded RNAs. you can take their genetic material, shoot it into a host cell, and get out virus. Because in some cases, like norovirus, a linear genome like this one, it also functions as the mRNA. It produces a polyprotein. And that polyprotein can be cut into smaller sizes and now begin to replicate the gene, plus single-stranded RNA that has been inserted into those cells. This is not possible with negative single-stranded RNAs. So I think, Sabrina, you might have been the one, I can't remember who, but I think it was you that asked me a question about, one of the review questions focuses on, hey, you infect with two different viruses. One has a plus single-stranded genome, one has a negative single-stranded genome. All you find in the end are that you get plus single-stranded genomes. Why is that? This is the exact reason why. So every other RNA virus, double-stranded RNA, negative single-stranded RNA, they have to have the polymerase actually in the capsid when they're being delivered into the host cell. Plus single-stranded RNAs don't need to have that. Annie?
So if you wanted to transfect like a negative single-stranded RNA, would you also provide the polymerase?
You would have to. So probably the way you would want to do it is you would provide the negative single-stranded RNA genome. You would also provide a plasmid expressing that RDRP, or put it into cells that already stably express the RDRP. Go ahead.
If you knew that the virus was a negative single-stranded RNA virus, could you do? I mean, I suppose maybe it wouldn't work for that one. I was going to say, could you put in the positive single-stranded RNA version of it?
Yes.
Okay.
It's possible. It's possible that you could do that. You probably have to know what that positive single-stranded RNA structure looks like, but I think it would be plausible to do. Yeah. Okay. Does this make sense, what they're doing here, and why this works for plus single-stranded RNAs? Okay.
Is it always making that endosome, or does it just fuse sometimes?
See, that's a good question. I thought it was a fusion event, but apparently I'm wrong about that. I don't know the answer. But somehow it gets in. It's magic, like most science. All right, so apparently the figure that started driving fits to everyone. Let's get two things clear. What's alpha CD? Antibody. Alpha oftentimes is used as a symbol to mean anti or antibody. So this is an anti-CD3001F antibody. What's SCD3001F? Recombinant protein. It's a solubilized version. In other words, it contains all the protein but the transmembrane domain that holds it into the cell membrane. Okay. So, I think before even looking at this figure, what we need to do is we need to go ahead and model what's actually going on in these two experiments so that we can understand it. I am one that, I think I've said this before, but I'm one that learns probably best by drawing things. If you're somebody like that, feel free to kind of try to model or map some of this stuff out as you're reading through the paper, okay? So what they end up doing is taking I'm just going to kind of draw a cell surface here. Oh, that's not the right tool. Cell surface. So here are the BV2 cells, the mouse cells that they're using in this experiment. And these are expressing on their cell surface. We'll draw it as a square. Here's our receptor, our CD3001F. Okay? In one experiment, they add antibody that binds to this. They also add the virus. And this is norovirus puking because that's what norovirus leads to. Okay. Bleh. Will it get into the cells?
No.
Why not?
The antibodies block.
Oh, perfect. So in this experiment, what they're basically running is, there are two different versions of what we would call competition assays. In this particular one, with the antibody, what they are doing is they're determining if they block the receptor with antibody, will it prevent entry of the virus, attachment entry of the virus into the host cells. Okay. Now they're using, in this experiment, they're using increasing concentrations of the antibody. So they run it numerous times. They try one with like no antibody, then they try 1 micromolar and 10 micromolar and 100 micromolar, and they'll generate this really nice curve showing, look, as that concentration increases, there's increased blocking of viral replication. Okay. In the other experiment, again using BV2 cells, again expressing this CD, but then they've got the solubilized form. What happens here?
The virus binds to that soluble.
Bingo. Yeah. So we would call this actually, this soluble, there's a couple of terms we might use. One, it's a decoy. It mimics the wild-type CD 3001F, and by binding to norovirus, the other thing we might say is it neutralizes norovirus and prevents it from binding to host cells. Okay. So two different ways to protect cells from infection is the goal here. Go ahead.
Can we go over ectodomain?
Yeah, we can try. Have you ever seen Ghostbusters?
It's been a while, I guess.
Okay, so they used to have this ectoplasma cooler drink, high C, like, yeah. So the ectodomain basically is no. The ectodomain really, in this case, is referring to the extracellular domain of the protein, or the region that would be on the surface outside of the cell.
That's actually binding.
That the norovirus would actually bind to. Yeah, that's what. Now, the ectodomain is a term I haven't heard used all frequently. I would probably more expect to hear extracellular domain, but it's really the same thing. Annie.
What's the benefit? Because they're pre-treating the virus with this. They're not exposed to the cell yet. So what's the benefit
point.
of just using the extracellular domain then? Why, like, is it more stable on its own?
I think so, and I think all they're trying to show is that, look, the norovirus binds, this would be what we call like a neutralization assay. What they're trying to show is that, look, it binds to this, and by binding to it, it inhibits its ability to bind to the host cells. That's kind of the overall theme. It'd be interesting to see if, I mean, you're right that they do end up pre-treating with it beforehand. It'd be interesting to see what happens if they didn't pre-treat. Actually, that's what they do with the mice, I think, if I'm not mistaken. Jonah.
Because it's like a cellular membrane protein, I'm assuming that part of it is non-polar, so maybe it would, like, if it had the portion that went into the membrane, it would maybe bind to the membrane.
Yeah, and so they've gotten rid of it, and this does not work with every this does not work with every transmembrane protein. There are some that can't hold their three-dimensional structure if you get rid of that transmembrane domain. This is just one where it seems like it really is almost like two separate domains or two separate motifs holding it together. Let me just skip to this figure real quick. So the summary of this figure, if you take a look at it, increasing concentrations of the antibody or increasing concentrations of that soluble product will improve cell viability, meaning what? What's the alternative way to look at that? Or what's another way to look at improved viability?
No infection.
Not infected, exactly. The cells aren't dying because they're not being infected. That at least is a reasonable interpretation of those results. Okay. I'll tell you what. I'm not going to grade critical thinking over the weekend, or the lit review over the weekend. Make changes if you want to. Go back, take a look at it again, make some changes. All right? Come back on Monday, though, ready to finish talking about this, because we'll try to finish the rest of it as we are going in and out of the lab. Have a great weekend, everyone.